Thank you. Today I'll be discussing a strategy to identify new symbiotic genes in the model legume Medicago truncatula. Below our feet lies a world of plant beneficial microorganisms like mycorrhizal fungi and nitrogen-fixing rhizobia. These microorganisms help a plant meet its required nutrients of phosphorus and nitrogen. While mycorrhizal fungi associations are prevalent among the majority of land plants, nitrogen-fixing rhizobia is limited to legumes. Symbiotic associations begin when signals are released in the soil and perceived by receptors, initiating a common signaling pathway in plants that activates known symbiotic genes. These genes are known as DMI2, DMI1, and DMI3, which stands for does not make infections, and they play a role in what is called early nodulin. Now, down the pathway, early nodulin is expressed, and I'll be referring to this gene as E011. The final response to the signaling pathway is nodulation and arbuscular mycorrhization. Now, since both of these share the common signaling pathway, and this pathway is conserved in majority of land plants, it opens this exciting possibility of manipulating this pathway into non-legumes. However, currently there are gaps in our understanding of the common signaling pathway. In addition, a classical mutagenesis-based forward approach is saturated, meaning that we are no longer finding new symbiotic genes from this approach. In response to this problem, we aim to utilize and optimize a genetic strategy known as suppressor screening to identify novel genes in the model legume Medicago truncatula, which is very similar, close relative to alfalfa. After identification, we aim to characterize the regulators of known symbiotic genes to connect the missing links in the common signaling path. Above here is a picture of Medicago nodules that show the phenotype NOD+. In a classical forward genetic approach, we take wild-type plants that show that phenotype, NOD+, which means that the genes are always turned on for nodulation. We then mutagenize and screen that mutant population, and from that screening, we isolate NOD- phenotype mutants. In contrast, for genetic suppressor screening, we take symbiosis defective mutants that are NOD-, mutagenize them, and screen that mutant population, and from that screening, we isolate NOD+ mutants, also known as suppressors. And in both of these approaches, it ends in further characterization of mutants. Below is a depiction of our research design that we developed and optimized for the genetic suppressor screening. We started with seed extraction and sterilization to get rid of any contaminants. We then began to germinate the seeds on agar plates, and one day after germination, the seeds were planted in a tray of turfase, which is a soilless media, and it's very similar to what you find in the inside of a baseball diamond. 10 days after planting, we inoculate with rhizobia to induce nodulation. And 5 days after that, we inoculate again with rhizobia to target the E011 expression. In total, 17 days after planting, we remove the roots from the tray of turfase and screen both for E011 expression and nodulation as well. And this is done both through a colorimetric assay as well as a nodulation assay. In the previous slide, I mentioned two phenotypes that we screened for, nodulation and E011 expression. In this picture below, you can see those two phenotypes mentioned. We have nodules on the left side and E011 expression on the right side. Above is a summary of my screening data. This is an ongoing project that many students have worked on. In total, we've screened 25,000, 22,000, and 23,000 mutagenized seeds for genes DMI1, 2, and 3 respectively. My contribution is solely to DMI3, where I've screened a total of 6,000 seeds and found 11 putative suppressors for E011 expression and 2 putative suppressors for nodulation. With our results in mind, our next steps and what we're currently doing is confirming those suppressor phenotypes that we found in the M3 progeny. We would also like to do a series of genetic inheritance and tests, aliasm tests, and a detailed phenotype characterization. In addition, genetic mapping of the suppressor loci and next-gen genomic sequencing is in the future. To conclude, understanding the mechanisms behind the signaling pathway will help identify potential targets to engineer more efficient nitrogen-fixing associations in crops that currently don't benefit from these interactions. It is important to note that our efforts have the capability of increasing the sustainability of agriculture, reducing environmental effects from nitrogen application, and decreasing the costs incurred by farmers towards the purchase of fertilizer. I would finally like to acknowledge and thank my mentor, Dr. Muthu Venkateshwaran, and Muthu Lab members, as well as thanking the WiSys Foundation for allowing me to present my research here at this virtual symposium. I'd also like to thank all the listeners for allowing me to speak. Thank you.